Okay, I think we can start. So it is my great pleasure really to introduce today Dr. Vinicio de Jesus Perez, who is a professor of medicine at Stanford University, and he's also the associate dean of MD admission also at the Stanford University. Vinicio was born and raised in one of my favorite islands in the world, which is of course Puerto Rico. I have great memories on Puerto Rico. So he obtained his medical degree from the University of Puerto Rico. He then completed an internal medicine training program at MGH in Boston and a fellowship at University of Colorado. He then moved to Stanford in 2004 as a postdoc in Dr. Marlen Rabinovich lab. And he's then, since then, the last 20 years, he was promoted to the rank of assistant professor in 2011, associate professor in 2018, and full professor last year. Vinicio's research focused on understanding the genetic, molecular, and cellular mechanism involved in the pathogenesis of pulmonary arterial hypertension. He's particularly interested in exploring how the Wnt signaling pathway affect the behavior of pulmonary artery endothelial cells, smooth muscle cells, and pericytes in PH. Vinicio is one of the most recognized leaders in the field of pulmonary hypertension. He has made very important contributions to the field. He published more than 140 articles in high impact factor journals like Circulation Research, European Respiratory Journal, the Journal of Clinical Investigator, Cardiovascular Research, the American Journal of Respiratory and Critical Care Medicine. Vinicio received several recognition and awards. He has a long list of awards, so I will not go through them. He's on the editorial board of a number of high-impact journals, including Circulation Research, the American Journal of Respiratory and Critical Care Medicine, and the Pulmonary Circulation. He's a member of American Heart Association Vascular Biology and Blood Pressure Committee, and he's also a standing member of the Pulmonary Vascular Disease and Physiology Study Section at NIH, where we frequently meet since 4 or 5 years. So Vinicio, we are really very happy that you accepted our invitation, and I look forward to your presentation. So please join me in welcoming Dr. Vinicio.
Eight buttons, yeah. There you go. All right. I should be on. There you go. Hi. All right. So thank you so much, Jacine, for inviting me. It is a true honor to be here with all of you today. So today I'm going to tell you one of the stories that my lab has been working on for the last 15 years. As Jacine pointed out, I've been interested since being a postdoctoral fellow in the Wnt signaling pathway. So today I'm going to share one of the stories that has evolved from this work. Before I move with the science, we need to talk about the clinical syndrome of pulmonary arterial hypertension. As a physician, this was my gateway into vascular biology. This is a disease that affects predominantly women. It is characterized by abnormal increase in pulmonary pressures, which leads to right heart failure and ultimately death. The good news is that despite being a rare disease with 120,000 people affected in the United States and Europe, we do have 16 medications, which for a rare disease is actually quite amazing. Unfortunately, they do not prevent the progression of the disease. So this is where my work focuses, trying to figure out how to untangle the molecular basis of the disease. Now, to tell you a little bit about what happens at the level of the pathology, you'll see this is essentially what a normal pulmonary vessel does. This is the airspace, alveoli. This is the vessel. You can see it's quite thin, allowing oxygen, gas exchange very quickly, right? So When you have pulmonary hypertension, what happens is you have an evolution where the lumen begins to get narrow. And what you can see is due to an expansion of vascular wall cells. Here you have the smooth muscle cells, you have the endothelium. And as you evolve, then you start seeing neointima formation. And ultimately, you see what is called a plexogenic lesion, which is filled with endothelial-like cells as well as other cell types. No surprise that if you start looking at the distribution of vessels as the disease evolves, you start seeing a rarefaction. And this is as a combination of loss of perfusible vessels, as well as the inability to regenerate new vessels. And this is where the focus of my research is. Why do we get these lesions? Well, this is where we should be. This is where we are. How do we get back here? Well, there are many mechanisms that are associated with the condition. Inflammation, drugs and toxins, endothelial apoptosis, mutations, DNA abnormalities. There are more here, as Dr. Zassi can attribute. And then the one that I focus on is on dysfunctional angiogenesis. Now, you can see that when the endothelium is injured, the thought is that these cells suddenly starts releasing factors, and through cell-cell interaction, it can influence other neighboring cells, activate leukocytes. These cells can transition into smooth muscle-like cells, fibroblasts. So you get a perfect storm that ultimately leads into that remodeling. So figuring out what triggers endothelial dysfunction could lead us to understand the ways that we can hijack this process and revert it. So we go from this to something like this. Now, before we go, we move in, the first thing was to try to understand a little bit better these lesions. So we have been limited for many decades to two-dimensional histology. So one of the things that we did in the last couple of years, partnering with Dr. Tran Lomark in Lund, was to use synchrotron-based phase contrast micro-CT, where you can actually take a slide, and using a synchrotron, you can actually get electron microscopy style images, which provide unparalleled access to these tissue sections and understanding the pathology. So from this that you can see here, we can actually see, and you can actually tell that this does not hint at the complexity of these vessels. You can see this is the pulmonary artery, and you will think that this lesion is only limited to the inside of the vessel wall. But what you can see here is that these lesions can expand and ultimately create a significant disruption. But not only that, you can even identify through this process 3 different kinds of plexogenic lesions, right? So you can see there are some that are your typical obstructive lesion, right? And this is a collateral trying to, get blood into the airspace. You can see these lesions that actually seem to be associated with bronchial arteries. So they arise from the systemic circulation. And then you have these lesions, which, as I told you before, actually apparently arise from the vasovasorum. So what I'm pointing here, all of these things have in common is they hint at dysfunctional angiogenesis. So the understanding being that the endothelium is certainly responding to something, but for some reason that endothelial cell does not know how to put itself into a new blood vessel. So before we can think of ways to target the process, the first place is understanding what it takes to really trigger these changes. Now, for those of you in the space of biology, you probably are all familiar with the VEGF signaling pathway. And again, this is actually a master regulator, which as you can imagine, is responsible for driving angiogenesis in every vascular valve. It's not the only one, as I'll show you, but it's the master. So back in the early 2000s, Dr. Focal and Dr. Tudor, who are two of the leading researchers in this area, identified, looking at these lesions, that there is an increase both in the VEGF receptor, as you can see here, and in the VEGF ligand. So ligand and receptor being up. Now, looking at this data, the hypothesis that is posed is, well, it's all due to an excess of VEGF signaling trigger an excess of angiogenesis. That makes sense, except that when I was a postdoctoral fellow, we started taking cells from patients. We will take the whole lungs and we will get the cells and we will culture them. And what's really interesting is that if you expose these cells to VEGF-A, the healthy donors actually form tubes. This is a matrigel assay, and you can see that the endothelial cells organize themselves into tube-like. So they know to put themselves into tube structures. The PAH cells, however, despite giving them VEGF-A, they do not respond appropriately. So it's almost like, even though they have the components, as I show you in the pathology, they don't seem to be responding. So, and interestingly enough, if we dig a little bit deeper, you will see that while you got cells that are responding, so this is the early stage of a tube-like formation. You go from, you have what is called the pathfinder or the tip cell, which has this filopodia, which are like radio antenna that are sensing the signals like VEGF. So they follow it, and as they follow it, the other endothelial cells follow, and that's how a tube is formed. That's called sprouting angiogenesis. The PAH cell, what you can see is that it forms less filopodia, but from there, there's not much movement, and this comes from live imaging time lapse. Going even further to electron microscopy, again, you can see that it's quite evident that the tip cell is forming these filopodia. You can see them here sensing the media, and the filopodia are sort of truncated. So what this shows you is that despite providing a steady level of the agonist, the endothelial cells are not sensing the agonist. So what is happening here? Well, it seems like you cannot just depend on VEGF. I mean, as such a powerful trigger of angiogenesis, you can imagine that there have to be compensators to really fine-tune this signal so the endothelial cells can carry out the work in a homeostatic fashion. And there are many pathways that are involved in allowing the cells to find each other, breaking the matrix so that they can expand, and then also recruit pericytes and smooth muscle cells to the wall. Among these pathways, we focus on the Wnt pathway. This was part of my postdoctoral work, and that's why I had this bias, because there's not much known about how Wnt, at least in the pulmonary circulation, how it interacts with endothelial cells to trigger these changes. So my first work was, what is the role of the Wnt signaling pathway in terms of interacting maybe with VEGF to regulate lung angiogenesis? Obviously, I have to start by explaining to you what Wnt is. Wnt is not just a pathway. It's a series of pathways with critical roles in development, stem cell differentiation, incredibly powerful and conserved through evolution. The one that most of you will know about from your molecular biology graduate studies is the canonical pathway, where a Wnt interacts with receptors and triggers the accumulation of beta-catenin, which then accumulates, goes to the nucleus, and triggers transcriptional genes. There's another pathway that controls calcium homeostasis in cells, and it's, of course, critical in development, particularly in xenopus, for example, which is where Dr. Randall Moon discovered it. And then there is this guy in the middle, which is the one that I've spent the last 15 years working, which is called the planar cell polarity pathway. What makes this interesting pathway is that This one controls cytoskeletal rearrangements as well as some transcriptional activity that allows the cell to understand its environment. It helps the cell communicate with others so that they can actually get together and form a structure, whether it's a bronchial tube, whether it's a kidney tubule, you name it. So when you think of a tube structure, the Wnt-PCP is likely involved. And there, the Wnt's ligands interact with a receptor called FRISL, G-couple receptor, and a tyrosine kinase called ROR2, which I'll tell you more about that. And then trigger changes that regulate cytoskeletal rearrangements through direct contact with actin, RO-RAC, and also trigger junction signaling, which changes transcription. So this was the pathway that we were interested, because again, if you think about it, endothelial cells need to communicate with each other in order to form a tube-like structure. That's what a blood vessel is. So to really understand how, how Wnt works here, the first thing is, well, there's 19 different Wnt's known. So which Wnt's are really involved here? So we use a very simple model. This is a tissue culture plate. We actually allow endothelial cells to grow in confluence, and at that point it's quiescent, right? They only grow in one layer. And then we actually had semi-confluent, where you can see endothelial cells are actually working their way to try to cover the gaps. And this is kind of an angiogenic state. So we harvested cells and we compared the RNA expression profile for all Wnt ligands in these cells. And what we found is that among the most expressed Wnt ligands, the one that really popped up was this one called Wnt7a. What's really interesting is that when you compare healthy, this is healthy, against PAH, there's a significant difference in the ability to express Wnt7a, and that is really interesting. So we decided to look in cells lysase, and we saw that the Wnt7a protein expression there was significantly reduced in PAH patient endothelial cells. Moreover, we decided to look further into tissues that we harvested from this patient. This is healthy donor. And again, lectin identifies the endothelium. This Wnt7a, you can see that is nicely expressed in endothelium as well as other cell types. Particularly, you'll see these epithelial cells here, which are labeled by RAGE. But when you look at PAH, what you see is a significant reduction in the signal of Wnt7a across the entire lung, but especially inside the vessels. You can see the abnormal anatomy. So all the evidence points to a significant reduction, if not absence, of Wnt7a in PAH endothelium. We went further because angiogenesis takes place in a three-dimensional environment, so we have to simulate a three-dimensional environment. So for that, we use an invasion assay pioneered by Dr. George Davis, now in Florida. You see the cells on top of the gel. The gel has VEGF-A, Wnt7a, or both. And you allow the cells to go inside, and you can see that they actually recapitulate sprouting angiogenesis. So using this, we were able to show that under VEGF-A, again, your healthy donors have nice tip cells, and you can see the endothelial cells behind forming a lumen, right? The PAH, as I've shown you in the two-dimensional images, again, they do elongate, but there's no tip cell there. However, if you add Wnt7a, if you piggyback Wnt7a to VEGF, again, with healthy donor, you can see that there is the amount of angiogenesis is greater. And in the case of the PAH, you start seeing tip cells. So adding Wnt7a to VEGF in the context of PAH endothelium can potentially rescue the sprouting angiogenesis. So the next step here is we did an organoid assay. Now, these assays many of you are aware of in the context of neurobiology and lung development. So we essentially, the way it works is that these little balls here are made of pericytes and endothelial cells. This is healthy donor with healthy pericytes adding VEGF. You can see that in the context of 24 hours, you can see sprouting coming nicely from the periphery. Again, the PAH, despite VEGF, there's not much. At Win7a, and as you can see here, there's even more sprouting, consistent with the 3D data I showed you previously. And again, if you add Win7a here, guess what? You can improve the level of sprouting angiogenesis. So this is evidence that VEGF and Win7a need to partner up in order to promote lung angiogenesis. This is all in vitro. So in order to test this in vivo, we generated a Win7a double flux using a endothelial specific driver. So you give tamoxifen to these animals, you take out Win7a in the endothelium. We used two models, one of chronic hypoxia and one of Sugen hypoxia, which Dr. Sassi here has also established very well. So using these protocols, we decided to measure pressures, look at pathology, and changes in the right ventricle. So to our surprise, we found that in both hypoxia and Sugen hypoxia, there was absolutely no difference between your Win7a knockouts and your wild types. Nothing. So this was disappointing, but at the same time, it opened a window here, like, why is this not happening? So we then look at the tissue. You can see again lectin endothelial cells. This is your knockout, this is your wild type. And you can see that in this case, you can see that Win7a is nicely expressing endothelium as well as other cell types. Emphasis on other cell types. The endothelial knockout really worked very well, right? But you can see here that there still remains other cells that are actually expressing Win7a. These cells, we believe, potentially are releasing enough Win7a to compensate for the absence of Win7a in the endothelium. Again, if you look here in hypoxia, you can see that, again, even though there was not much Win7a in the endothelium, there was an increase in Win7a in these cells here. So what we hypothesized was that there's a source that is compensating for the absence of Win7a in endothelium. So we use available single-cell RNA-seq maps now. This is from a publication by Shoup in 2021. Real good research if you're really interested in looking at genes. And guess what? The cell that expresses the most Win7a in the human lung and in the mouse lung is the alveolar type 1 cell. You can see type 1. These are the cells that really line up the inside of the alveoli, and is the cell that interacts closely with endothelium as part of the endothelial alveolar capillary barrier. And this is again showing you how the type 1 here is expressing a lot more Win7a compared to other cells. So then we decided, well, maybe what we need is to get rid of all Win7a in the animal. So we did a heterozygous or a homozygous under a ROSA-CRE driver, which hopefully gets rid of all the Win7a in the lung, including the type 1 cells. And again, for this one, we limited ourselves to a hypoxia model and repeated. This is a heterozygous animal, and this time around, we were able to show that in the setting of hypoxia, pulmonary pressures measured by right ventricular systolic pressure went higher in the animal. The level of right ventricular hypertrophy was greater. And when we isolated endothelial cells from these animals, again, you can see that with the heterozygous compared to the wild type on their VEGF-A, which creates really nice vascular networks. Again, like the PAH cells that I showed you earlier in my presentation, did not do so. But add Win7a, and guess what? You get some recovery of the angiogenic behavior. I'm showing you data for a Win7a heterozygote. What's interesting is that when we do a Win7a full knockout, these animals die 10 days after we give them tamoxifen. And what's really interesting is that when we look under the microscope, we see a complete breakdown of vessels, which is really interesting. And it will make sense as I show you some of the data subsequently. But we came up with the following model. We believe that what's happening here is that pneumocyte and endothelial cells are producing Win7a that then interact with the Wnt receptors together with VEGF receptor 2. What we find is that these two receptors may be interacting and potentiating the VEGF signaling by directly modulating phosphorylation in the cytoplasmic domain of VEGF receptor 2, which ultimately allows the signal to follow and trigger angiogenic responses, motility, cell-cell alignment, tube formation, which are important to be able to preserve the pulmonary vascular bed. Now, this is all well and good, right? And this opens a lot of questions that we're exploring. But the one I want to focus for the remainder of my talk today is, what is the role of the Win7a receptor here? Who is that? Well, I already hinted at you earlier that the receptor for Win7a is a tyrosine kinase called ROR. Well, the first thing we did using our semi-confluent assay is we look at expression of Wnt receptors, and across all non-receptors, again, the one that goes up in semi-confluent cells, which are, again, remember, expanding, is ROR2. And that is not seen in the PAH cells. Likewise, look at the expression pattern is significantly reduced in PAH patients, which parallels the drop in Win7a that I showed you earlier here. If we look at tissue again, and this is actually the advantage of having a biorepository where we can actually look at this, you can see again, ROR2 is really nicely expressed across the whole lung, but particularly in the endothelium, which is the focus of our interest. And again, when you look at PAH lesions like this complex plexogenic lesion here, there's barely any ROR2. No Win7a, no ROR2. You can see where this is going. But remember that our concern was, well, if you need VEGF and Win7a together, likely these receptors potentially need to be interacting together, and that may be why this crosstalk is important in angiogenesis. So we use what is called a proximity ligation assay. If the two receptors are together, we use an antibody engineered so that when the two receptors are together, you trigger a signal. Each dot here represents a complex between VEGF and ROR2. With VEGF-A alone, you can see that there are some complexes present in these endothelial cells. With Win7a, again, you see the complexes. But it is when you add both, you can see that the number of receptor, VEGF, ROR2 receptor complexes significantly increases. Moreover, when we look at the status of VEGF receptor 2 phosphorylation in residues that are critical for its activation and signaling activity, you see that using VEGF stimulation over 60 minutes, there's a clear activation in healthy donor, in cells transfected with a control siRNA. But if we knock out ROR2 with an siRNA, you can see that we can prevent the phosphorylation of these critical residues, leading us to conclude that ROR2 is critical for the activation of VEGF. So ROR2 primes VEGF receptor 2 so it can properly respond to the signal. Okay, so yeah.
So two questions. One is that, I just want to kind of understand correctly.
You have reduced Wnt7 expression in the pulmonary hypertension lungs, but you also have reduced the receptor. So both happened, right? That's kind of when you take the tissue from patient, that's late.
Yes.
So my question is that you did the global inducible knockout of Win7A, is there a model of AT1 Cree allow you to dissect that? And then the next question is the row 2 is also reduced. Is there any hope? Because these two both happened. Now you don't have Win7, you don't have a receptor. So how are you going to reverse the process?
So first question, we have developed a HOXP1 animal model, which allows us to knock out Win7A in epithelium. And we are currently conducting those studies. It's taking us a bit of time. This animal, as you can imagine, is finicky, but stay tuned. For your second question, what's really interesting is that when we add Win7A, we still can get a rescue. So even though, so at this point in time, what we have shown is that when we do Win7A, we can increase the expression of the ROR2 receptor. So that's really interesting. And it may be also, if you add VEGF stimulation, you can also trigger activation. We also see that HIF signaling, I'm not showing you this data, but HIF signaling, it triggers not only VEGF expression of VEGF ligands, but also Wnt and ROR2. But what we have found is that there is a Dr. Jekyll and Mr. Hyde effect, where HIF2 suppresses ROR2 Win7A, HIF1 promotes it. And not only that, but there's also significant epigenetic repression around this. So what we have been doing is, by reversing epigenetic repression and by inhibiting HIF2A, we've been able to restore some of this activity. But ultimately, I think what we have found is that if you add Win7A, there may be some residual ROR2 activity, but together with VEGF signaling, it may be enough to trigger expression of enough ROR2 and Win7A to allow some. But here's the thing, as you appropriately point out, we are using tissue from patients with end stage of the disease. Ideally, we will want to see how this evolves, because there may be a window of time where you can do this at an earlier stage. So what we are testing right now is, we have in our hands a HIF2A model, which develops pulmonary hypertension tremendously. And there's no Win7A there, because remember, too much HIF2A, it shuts down Win7A. So we're giving exosome carrying Win7A particles to see if that could allow us to rescue that model. So that work is currently underway.
Thank you.
My pleasure. Yes.
When the Win7A signaling is happening in the context of VEGF, is that for ongoing turnover of endothelial cells to maintain the vascular branches, or is this, some sort of novel angiogenesis that's happening in response to whatever?
I think it's the latter. And unfortunately, due to the lack of human tissue at earlier stage, we don't have. However, what I can show you is that in the context of early hypoxia, so what we did is, in this same paper, we took mice across 21 days, and we sampled them 1 day, 7 days, 10 days, 14 days. What's really interesting is that the level of Win7A goes up very early, along with VEGF. But then by the time you get to 21 days, which is end stage, it's back down.
That suggests that in the disease, there's some sort of predisposing disruption of the vascular bed that requires a regenerative response.
Yes, sir. And that's kind of the leading model right now for dysfunctional angiogenesis. However, we don't know how it starts. We don't know how. We talk about gene environment, but the truth is, I think you're pointing out the crux of the field right now. Like, okay, that's great. The endothelium is sort of like, ground zero. and it leads to all of this. And just like in the context of fibrosis, like there's an inciting event, but we just don't know what it is and how it triggers and what pathways are responsible. I will propose that this is a pathway that is important at that early stage.
Endothelial cells.
Yes.
They're only endothelial cells. There's no other cell type.
No, so I think this is the question I was referring to, because the key cell that expresses Wnt7a is the alveolar type 1 cell. So this cell also is very responsive to hypoxia and triggers Wnt7a just at the same time as the endothelial. So it seems like this crosstalk is critical. So I think the point being that the ideal study, as we were talking about, dissecting how Wnt7a regulates epithelial cell behavior, but also that crosstalk, I think that's something that in this particular study, we're hoping to take these models and do some single cell analysis so we could dissect more cell-cell communications to receptor ligand. But again, the field is wide open. These are all important questions.
I have a quick question. Apologies, I don't know about PhD.
No, please.
PhD. So there are like 14 different wings.
19, believe it or not.
Okay. So, there's a lot of redundancy also in the wings.
Yes.
So I'm very surprised that there is no another wing that kind of pop up and that's the job of Wnt7a. But I wonder, do these patients, they lack the expression of Wnt7a from the very beginning of their life? I mean, or is something that kind of comes later? There's a compensatory effect from other Wnt signal, from other Wnt molecules until this becomes an issue?
I wish I could answer that question. That the answer, at least in humans, since that's where you're alluding to, is due to the fact that we don't have access to tissue until they reach the end stage. We cannot answer that question. I can tell you though that in the large genomic studies that have been done, where full gene sequencing has been done on patients, over 1,000 in the UK, here in the United States, also PH Biobank over 1,000. There's no mutations in Wnt7a or ROR2 that we can link. But what we have found is that these guys are exquisitely regulated by epigenetic mechanisms. Like the amount of histone DNA methylation that you find in these gene loci, especially, so we just did an ATAC-seq on endothelial cells on pericytes. It is amazing. So these are really epigenetically regulated. And we think that obviously that's not something that we can see with full genome. So I think beyond single SNVs like variants, et cetera, I think epigenetics play a role. Now, to answer your initial question, there's 19 wins. So why is this one important? So we have been looking at the Wnt-PCP pathway, and the other ligand that's the classic is Wnt5a. So we've tested Wnt5a, and believe it or not, these cells, it doesn't have the same angiogenic effect. But what's really interesting is if you give these cells other Wnt that are known to activate canonical signaling, what they do, however, is they don't trigger tip cell formation, but what they do is they trigger stalk cell, like the cells that form the body, but they seem to be more relevant for proliferation. But the fact is that if you don't have that tip cell arrangement, tip stalk, where the tip drives the direction of the growth that comes in, which the Wnt canonical controls, so you need these cells to be coupled. So the short answer is that in the great scheme of things, I think that these ligands can play a key role in repair, but eventually at some point, these other Wnt ligands may play a pathological role. And of course, we will love to look at that further. But again, this is why for these Wnt7a and ROR2 animals, we're doing some single cell RNA-seq, because now that we have this technology, I think we can get more, especially if we can do some time-lapse event. But yeah, I mean, the question is open. Yes.
Just on the cell crosstalk, is there much information on whether like macrophages play a role in kind of altering this as well, altering that signaling pathway as well?
Outstanding question. At least in pulmonary arterial hypertension, macrophages are key drivers of vascular remodeling. They crosstalk with fibroblasts, with small muscle cells, endothelial cells. You can even see in these lesions clusters of immune cells that organize into tertiary follicles. There is clearly crosstalk, and the single cell work that's been done so far in animals and humans suggests that. The role of Wnt signaling in that has not been explored. It's something that, you know, we haven't ventured in, but I will be surprised if it doesn't. I will be surprised. Any other questions? This is great. I love please continue with the questions. So this is now, so going back to ROR2, we then decided to take the same approach of knocking down ROR2 in endothelial cells and doing animal studies. Compared to Wnt7a, taking ROR2 out of endothelial cells does lead to worsening in pulmonary pressures, right ventricular remodeling, which you can see here in these echo data. This is the right ventricle, left ventricle. With in the ROR2 echo, the right ventricle is significantly larger and weaker when you look at fractional shortening. When we look at the number of vessels as well as the number of muscularized vessels that we see also a significant dropout of cells, of vessels under hypoxia and greater muscularization. So not having ROR2 in the endothelium seems to trigger a phenotype that's much stronger than that of Wnt7a. So for this one, since this is, you know, we did this at a time where single cell was much more accessible to us, we decided to take these lungs out and do single cell. For this, what we did is we focus, of course, on endothelial cells. And here, now we know that there are different subtypes of endothelial cells. As you know, we got general, aerocytes, venous, arterial, and at least looking at the numbers in the context of the endothelial knockout, what we see is that even at baseline, there seems to be a significant increase in the arterial in these aerocytes here. We don't know what this means yet. We're doing stereology to look at this further to see how this correlates with structural changes in these animals. But what's really interesting is that in our analysis so far of what pathways seem to be more altered in these endothelial cells under single cell, what came out is that there's significant disruption in VEGF pathways. We also saw changes, interestingly enough, in vascular permeability, matrix protein, basement membrane, and integrin binding. You can also see changes in extracellular matrices with receptor interaction. So this is interesting, right? Because after you complete your sprouting angiogenesis, the next step is you have to the vessel needs to mature. You need to form an endothelial barrier so that the blood that you push into that vessel doesn't leak out. So what this pointed out to us is that for some reason, not having ROR2 there seem to be altering pathways that are important for endothelial barrier integrity and maturation. So of course, the next step was to see whether the lungs of these animals demonstrated increased permeability. So for that, we took our animals and gave them Evans blue. Now, Evans blue, if you inject it, should stay inside the vessels, right? So you can see here wild-type normoxia, wild-type hypoxia. With hypoxia, there's some increase in permeability, which is thought to be due to the fact that hypoxia may be triggering new vessel growth, right? But look at the knockout. The knockout, even at baseline and with hypoxia, have a significant increase in the leakage of Evans blue. And this actually pointed out that indeed, in the context of having no ROR2 in the endothelium, there may be increasing permeability. So what regulates barrier integrity or vessel maturation, which is the second stage of angiogenesis? You need the coordinated action of endothelial cells interacting with the matrix to the establishment of focal adhesions, which allow the cell to sit and hold tight to that basement membrane, as well as forming endothelial cell-cell junctions, like junctional complexes, gap junctions, etc. Integrins were one of the pathways that seemed to be most altered in our single cell. Now, what are integrins? So for those of you, integrins are these receptor complexes that exist in inactive conformation. They usually are closed like that, but when they interact with matrix proteins like collagen, fibronectin, suddenly these receptors achieve an active position. And with that, they can actually trigger signal, and they are key for mechanotransduction, allowing the cell to hold tight. So we decided to actually focus on this particular heteroelement. Again, this is a huge, you can combine alpha and beta integrins in so many ways that ultimately you can go crazy. And it all depends. These receptors form in response to extracellular matrix protein. And the one that we were using was fibronectin to grow these cells, because that's one of the most abundant in the basement membrane where the endothelial cells reside. So we decided to look at beta-1 integrin. What is happening to beta-1 integrin as a surrogate for the focal cell dynamics in this cell? Is there a link between ROR2 and integrin beta-1 activation in endothelial cells? So first thing is, are integrin and ROR2 in some way interacting in the focal adhesion? So here is ROR2. This is a healthy endothelial cell here. You can see ROR2 is in the membrane, also in the cytoplasm. And integrins also, you can see, can be found in the membrane as well as in the cytoplasm. But also, you can see these comma-shaped structures there. Those are focal adhesions. They form these classic coma-like shapes. And when you look at ROR2 and ITGB1, there seems to be a nice colocalization here. Look at PAH, where again, ROR2 is reduced. But interestingly enough, if you look at the ITGB1, of course, there's less colocalization. But what was really interesting to us is that the number of focal adhesions in healthy was lower compared to that of PAH. So in PAH, there's an increased number of focal adhesions. So that's really interesting, right? How does that relate to the capacity of the endothelial cell to form a barrier? So we did a very easy assay. We took these cells and we do a FCCP dextran assay. You put the cells in a bucket, allow them to form a barrier, and then put dextran beads, and you count the number that make it across that barrier. Compared to control, cells where we knock out ROR2 have increased permeability. I told you also that these cells have increased focal adhesions based on what I show you. Also, we saw that here. So you will expect that more focal adhesions means that the cells will adhere strongly to this. So, and that's what we actually found. Interestingly enough, adhesion is greater. Again, knocking out ROR2 increases the number of focal adhesion, but not only that, it also correlates with a reduction in weak adhering, which we use to mark adherence junctions. So these cells, even though they're attaching strongly to the substrate, they have less permeability. So this essentially points out that ROR2 seems to have a link with the focal adhesions, which ultimately change the way the endothelial cells are interacting with the matrix and with each other. Of course, you want to know what happens to ITGB1 in a patient's cells, because you will imagine, well, if ITGB1 is increased in the context of ROR2 and ROR2 is down in PAH patients, ITGB1 should be up. And effectively, when you look at the lesions, we're looking at levels of active ITGB1. This is the active integrin. You can see that levels are significantly increased, and not only in endothelial cells, but across the different cell types. So integrin seems to be playing a key role here beyond the endothelial cells, but particularly in the endothelial cells. Focal adhesions are important in mechanotransduction. So when the focal adhesions are established, the cell generates force against the matrix. So if you have more focal adhesions, that doesn't necessarily mean that the focal adhesions are functional, that they're generating force. So what we did, we used a FRET sensor. We transfected the cells with this FRET sensor, which allows us to see how much force in micronewtons each of those focal adhesions generate. And again, control here, you can see signals in the periphery. Again, you can see the structure of the focal adhesions in these coma-shaped structures. Taking ROR2 out, there's more of these. And not only that, but these integrin focal adhesions are generating significantly greater force against the substrate. We also repeated this in PAH endothelial cells and found the same. So the next step we did was we actually transfected a ROR2 expression construct with the hypothesis that by restoring ROR2 levels to a physiologic value, we should be able to restore the functional capacities of the endothelial cells. And as you can see, we were able to show that in the context of ROR2, PAH cells were able to recover their adhesion, as well as permeability greatly improved. The level of FRET reduced, and apoptosis, which is interesting, this is actually one of the markers of cell survival, also improve in these cells. And I'm sorry, this should all have statistical significance, but basically being able to restore ROR2 is able to return physiological function to these cells. Okay, so how does integrin regulate integrin activation? So this is an important question. So as I told you, integrins are activated by interaction with the extracellular matrix. This is called outside in. Outside, here's the matrix. In, where the receptors cluster and trigger signaling through two tyrosine kinases, SRC and FAHA. And this is important to promote survival as well as create the structure that is critical for barrier integrity, right? So this was our priority. Our hypothesis is that we are going to be able to see greater SRC and FAHA signaling, and that excess signaling was going to be more prominent in ROR2 knockout cells. But to our great surprise, when we take out ROR2, we actually saw that there was a decrease in phospho-FAHA and also a reduction, although not significant, in SRC. This is the total opposite of what we were initially thinking. So we decided to look at the structural analysis. And again, we found that with SIROR2, you can see an increase in the level of active integrin. But again, the phospho-FAHA was significantly reduced compared to control. So this is not outside the traditional outside-in signaling. So this puts us in a pickle. If I have active integrin, and I'm not triggering FAHA signaling, then how are these integrins actually working? Well, yeah.
We can keep a couple of minutes for questions.
Absolutely. So just to wrap up, there is inside-out signaling. And in this case, you actually need a signal that comes from inside the cell through thallium kindling that then activates the integrin from inside for adhesion. And this is where we actually found that taking ROR2 out increases tallying activity. So this allows us also to look at this in our PAH cells. The last thing I'll show you is that we decided to look at the RNA-seq profile of this siROR2 to see how is then ROR2 leading to the activation of integrin. And what we found is that among the most disrupted components was actually vesicular trafficking, particularly this guy here called RAP12. Now, RAP12 is important because it's important in recycling integrins. Integrins, when they are active, they need to be recycled and returned to the membrane in inactive form. That's how you keep things going. And if you don't have RAP12, then the integrins will stay in the membrane and will remain perpetually active. So no recycling, we feel that this leads to activation. So knocking out siROR2 led to a reduction of RAP12, which is consistent with our RNA-seq. And you can see this also at the level of the cell. And most importantly, you can see that in PAH cell, there was a reduction in RAP12 natively, again, correlating with reduction in ROR2. So what we did was we looked at the balance between integrin and tallying and RAP12 between cytoplasm and membrane. You should expect more integrins, more integrin and more tallying in the membrane in the setting of PAH because you have more inside-out signaling. And this is what we saw in PAH, also in siROR2 cells. And again, here you can see how in this setting, we decided finally to knock out RAP12 to see if we could phenocopy what we see when we take out ROR2. So again, this is in control. This is your integrin. This is RAP12. You can see that it interacts nicely with cytoskeleton and focal adhesions. And when you take out RAP12, look at that. You lead to a similar phenotype where you lead to an activation of ITGB1 in these cells with all the functional changes that you will expect. So the model we're currently proposing here is that ROR2 interacts with ITGBA and they regulate FAHA, which is outside-in signaling, and you need RAP12 to keep things fresh, to recycle. But in the context of no ROR2, you actually have increased clustering and activity that leads to a change from inside out to outside in as a result of no integrin recycling. And this leads to the disruption of the membrane and impair angiogenesis. To finalize, the model we're proposing is that Wnt7a and ROR2 and VEGF are important in the early phase of angiogenesis. But in the context of the second phase of angiogenesis maturation, ROR2 seems to have a more important role. So this is work done by my lab, particularly Dr. Agarwal and Ankita Desai. These are collaborations. Thank you so much for your time.
Thank you, Vinicio, for this fascinating work. Yeah.
Thanks, Yassine. Wonderful talk. I'm thinking about the potential translation of your results.
Yes.
And I used to work on Wnt7a many years ago. But, you know, one of the things I learned about Wnt's is that they're just famously hydrophobic and difficult to drug. So I'm also aware of the fact that Wnt7a has multiple isoforms. I think it's got a C-terminal isoform. And I'm wondering whether there's any potential for, you know, using or modifying that C-terminal isoform to drug the kind of approaches you're using here.
That's really interesting, and it's kind of timely because last year there was a report where the structure, the structure of the ROR2 receptor, the extracellular domain, had been crystallized, and they were able to identify particularly ways particularly the lipid moieties, which I think you're alluding to, that Wnt has, which are important for its capacity, that sites where you can potentially engineer Wnt ligands that may specifically interact with ROR2. Now, I'm thinking here that one way of doing this, so I'm not sure that using Wnt7a as a therapy is going to work. And I totally agree. This has dodged the cancer field for years. I think the key here may be targeting the receptor itself, and it may be targeting some kind of agonist, or finding a way to indirectly trying to increase the receptor. So rather than targeting the ligand, it may be more effective to targeting the receptor. Now, whether this could be using some epigenetic derepressors, some HIF modulation, some genetic way to overexpress ROR2, that may probably be the most straightforward way, rather than you trying to target independent Wnt's.
Have you tried to overexpress the receptor, for example, in the Susan hypoxia model? To see
Oh, that's a good question. We haven't on either rat or mouse. ROR2 is reduced in the rats, in Sujan and monocrotaline, and we are discussing this with colleagues like Yu-Yang Sao, who's developed this nice adenoviral that can specifically target endothelium, to try to test that.
So in your various knockouts, is there any disruption of initial branching morphogenesis of the vascular bed in the lung? So in other words, is the Wnt7a just for remodeling and all of the other mechanisms, or is this a developmental mechanism that then gets hijacked?
Great question. So as I alluded to, when we take Wnt7a out completely, the ROSA model of Wnt7a homozygous, the animals die in 10 days, adults. And what we find, we're looking at the histology, and now we have been doing also like vibrotome sections, the vessels just break down in the adult, which is interesting. So I cannot tell you about sprouting angiogenesis because the lung is really difficult to for this to study this, or So, but clearly it has a catastrophic effect. Now, if you look early, and you probably know the literature from Dr. Ben Bars on Wnt7a. Wnt7a is critical for brain vascular development. So if you take it out from development, the animals die. And also it has important role in branching morphogenesis of the lung. So the short answer to your question is, I think it does have a key role in the vasculature. That's as much as I can tell you. Is that role limited? I mean, here's the thing. The adult animal shouldn't have sprouting angiogenesis. This is an animal that's otherwise normal. I inject tamoxifen, and 10 days later, 7 out of 10 of those animals are dead, and the lungs look like muck. We've done a necropsy, and nothing else is that. So it seems like there's something unique to the lung in the adult mouse. So for the ROR2, we've done this. The animals are happy. We haven't done a global ROR2 knockout, so that will be interesting to do. One question that I've also been asked routinely is whether aging has an effect on this, and that's also something that I think it's worth exploring.
Thank you very much, Vinicio.
My pleasure. Thank you.